Welcome to the University of Manitoba Alumni and Friends Virtual Learning for Life series. This is our sixth session of a total of 9 weeks. My name is Tracy Bowman, and I'm the Director of Alumni Relations and a proud alumna, and will be the moderator for today. Thank you for joining us and making this event part of your day, and in general for choosing to stay connected with your alma mater in this way. We have been able to offer this program free of charge to all of our 145,000 alumni living in 140 countries around the world, thanks to the very generous sponsorship of one of our affinity partners, IA Financial. Many thanks to them. Delivering learning for life opportunities is a very important role for the University of Manitoba, and we're so proud that we're able to showcase many of our leading professors and researchers in this way. Now, just a few housekeeping details before I present today's speaker. You are viewing this webinar in a YouTube link, and you will see both of our presenter and as well as his PowerPoint presentation. Today's session, like previous sessions, will be recorded and posted on our website within about a day or so. You were also sent a link to Slido. That website is www.sli.do and the password VL10. And that's what we will be using to facilitate the Q&A. So I encourage you to enter your questions there, and then I will be reading them out to our presenter, and we will get through as many as we can. So now I'd like to introduce our speaker today, Dr. Jason Kindrachuk, who will be presenting on COVID-19, the emergence and spread of a pandemic in the age of social media. Now, just a bit about Dr. Kindrachuk. He's an assistant professor in the Department of Medical Microbiology and Infectious Diseases at the Rady Faculty of Health Sciences at the University of Manitoba. And he holds a Tier 2 Canada Research Chair in Molecular Pathogenesis of Emerging and Re-emerging Viruses. He has a broad expertise and interest in emerging viruses and outbreak preparedness in both developing and developed nations. His research focuses on the investigation of emerging virus circulation, transmission, and pathogenesis, and in particular, those viruses that pose the greatest threat to global human and animal health. He's also served on multiple expert committees with the World Health Organization, and he's actively participated in media outreach locally, nationally, and internationally. And you may see a number of these media reports citing Dr. Kindrachuk's expertise on Today. So with that, over to you, Dr. Kindrachuk.
Great. Thank you so much, Tracy, and welcome everybody. Hopefully everybody has not gotten too tired of hearing my voice or hearing my words about COVID-19, but that's what happens when you're one of very few virologists that are willing to still take media interviews during a pandemic. So, really today what I want to do is cover some basic information about COVID-19, but also talk a little bit about what we do know and what we really don't know at this time period, and where misinformation and disinformation kind of plays into this. In particular, since this is really the first pandemic where we have truly been in the age of social media, and where social media really predominates our ability to get news and updates and data. Whether you're a member of the general public or you're a member of the scientific research or clinical community. So with that, let's talk a little bit about viruses and talk about an emerging infectious disease. So this figure, I'm just going to put up my pointer for you. So this figure is from ProMed and the International Society for Infectious Diseases. And this was basically a report that was done from July to September 2019, really highlighting the different infectious diseases that were being battled or that were predominating in different regions of the world. Now, the size of the words and the location don't specifically define how large an outbreak is or the specific location. So Ebola here is designating that there is an ongoing Ebola outbreak in the DRC. Same thing with polio up into north central Africa, but it doesn't necessarily tell us what the overall toll of those diseases in the region is. But what we can see is that this picture is extremely complicated, and that gives us an idea of what we're battling as virologists, as researchers, as clinicians. Who focus on infectious diseases on a daily basis. Now we fast forward to 2020, and the period from January to March. And what we see is obviously the distinct change. COVID-19 has really taken over everything. But what I think we really need to keep perspective of and maintain perspective of is the fact that while COVID-19 is really what is predominating the news headlines and is on our minds, we are still battling infectious diseases across the globe, and in particular, the regions of the world that I tend to do a lot of work in, mostly in West and Central Africa. The DRC right now has two ongoing outbreaks of Ebola that are not related to one another. So when we think about just the sheer amount of stress that infectious diseases exert upon us, my statement to people is always that this is a battle that I don't think we are ever going to win. It's one that we are merely trying to subside for as long as we can. Now, this slide, I've debated pretty much for the past 6 or 7 months whether to continue to show it. In particular, because of the time period that we are currently in with the COVID-19 pandemic. But I think it harkens back to statements that many of us have been making for a long period of time. And for myself, I've been involved in emerging and re-emerging viruses for 13 years now. We constantly talk about the idea that we truly aren't ready for the next pandemic. And I think in a lot of cases, people have maybe assumed that we were, highlighting the doom and gloom aspect because that's where our research focus is. And in fact, WHO a couple of years ago actually put together a working group, which was the Global Preparedness Monitoring Board. And they were an independent monitoring and accountability body that basically was put in place to try and examine whether or not, as a global community, we were actually prepared for an incoming pandemic. And ultimately, what they found was we are not, and we were not. And most of that came down to 7 urgent actions that were identified that were critical for us to be able to combat a new pandemic. It was a virus that was reemerging or one that was newly emerging, and it came down to really 4 key points that, in my opinion, that I think are the most distinct. The first being that heads of government have to commit and invest. I do a tremendous amount of work across Africa and in areas that are completely resource limited. These areas, unfortunately, are the most vulnerable to emerging viruses and emerging infectious diseases. And they are really our weakest link in the chain to be able to battle these diseases globally. So, I think this is a really critical point for us to think about the fact that as a global community, and one that is interlinked, undoubtedly, in 2020, we need to be able to invest and build up the infrastructure and the expertise in the most vulnerable regions of the world if we ever expect to be able to combat infectious diseases as they emerge. Countries and regional organizations have to lead by example. There's no debate about that. We need to have countries in place that are willing to kind of lead the charge. I think Canada has done an immaculate job of that. In particular, my own department, medical microbiology, infectious diseases, has been doing this with HIV for decades now. And our department, in conjunction with the Public Health Agency of Canada, have been doing this, globally for other infectious diseases, including Ebola. But we also need other regions of the world, in particular, regions or countries associated with, say the G7, to be able to stand up and help lead the charge. All countries have to build strong systems, so we need to have monitoring systems that are in place, and we need to have some amount of coordination between those countries to be able to combat infectious diseases. And ultimately, countries, donors, and multilateral institutions have to be prepared for the worst. All of these points, I think we can go through and we can look at and basically put big red X's beside to say this is how we weren't prepared for COVID-19. And that's the unfortunate position that I think we found ourselves in. And we've said this for years now, coming out of SARS, coming out of 2009 influenza through the H1N1 pandemic, coming out of the West African Ebola epidemic, that we needed to, as a global community, really identify these areas of need and circumvent those. And I think we are still at an infancy in actually doing that. So what is it that I do, and where do I fall into in a picture of emerging infectious diseases? Really, what I'm trying to and have been trying to establish, whether rightfully or wrongfully, over the past 3.5 years now here in Winnipeg, and then during my prior time when I was still at the National Institutes of Health in Washington D.C., is really trying to integrate what we call One Health approaches to emerging infectious disease. And I know the slide is a little bit busy, but really when I'm talking about this idea of One Health, it's really trying to integrate human and animal health along with environmental health. And I know that when I've talked a little bit about environmental health in the past, people immediately, I think, start to think, are we talking just about, trying to preserve the environment? Are we just talking about climate change? Well, yeah and no, they're all interlinked. But what we have to think about is ultimately humans and animals, in terms of our health, are ultimately linked back to our environment. And what we know, especially with the types of viruses in infectious diseases that I work with, we know that there is a link in between contact of humans with animals that carry these diseases. And what we do know is that contact is basically being increased in a deleterious fashion or in a negative fashion by changes in our surrounding environment. I'm going to walk through a little bit of that. So when we think about this idea of things like climate change, ultimately, I think, again, we look back at the scientific community. I think pretty much everybody in the scientific community at this point predominantly agrees that climate change is occurring. But what we need to start to think about is how climate change is actually having an effect on our surrounding environment. And Dr. Ian MacKay from Australia had actually put this wonderful figure together. Ian is one of many friends that I have now gained on social media through Twitter. The other Dr. Kindrachuk in my household, who is I think vastly more talented than I am. I've told her many times that once I hit the age of 40, I'm at an age where I likely will not gain lifelong friends in person, but social media has afforded me that ability to make friends that I don't necessarily have to see all the time. And I think other people feel the same way. So that's fantastic. But Ian put together this amazing figure, and it really talked about the idea of how climate change is affecting essentially this interaction of us as humans or even wildlife with infectious diseases. And what we can think about is the fact that without going through all the specific examples, climate change ultimately is changing the dynamic of how we interact at the interface of infectious diseases. And really what we need to start to understand is how to essentially try to mitigate many of these interactions so that we can try and reduce the amount of infectious disease spillover that we're finding, but also to try and better understand what types of safe checks we can put in place to try and reduce the amount of spillover that we see. So, again, I'll refer back a lot to Ebola because that really is what got me into research in the first place. In the Democratic Republic of Congo right now, we know that there has been a second outbreak of Ebola within the country. And all data right now points to the fact that this is what we call a new spillover. So the virus has basically spilled over from wildlife, likely bats, into humans. Can we start to understand, how climate change is driving some of those interactions? Because what we're seeing ultimately is that many of these, what we call zoonotic infectious diseases, so diseases that spill over from animals into humans, what we're seeing is a general increase over the past couple of decades. And that increase correlates very well with climate change. And there's actually been some pretty good studies that have shown causation between those events. So I think we need to start to put those into perspective. And the reason why these things are important is that when we think about the idea of infectious diseases, and in particular zoonotic diseases that basically are spilling over directly from animals into humans, what we need to understand is that, first of all animals are not equal. And that's one of the things that really makes, I think, my area of research interesting, but also extremely difficult. So if we think about things like Ebola, and we'll talk about coronaviruses and COVID-19 pretty quickly in the talk, we think that these viruses basically persist in bats. And what that means is that bats are carrying these viruses naturally, but don't show signs of of disease or any signs or symptoms of illness when they're infected. So we refer to them as basically being a reservoir. The problem with this is that bats essentially are kind of nature's perfect reservoir. I love bats. I think that they're amazingly cute, especially the larger fruit bats. But they carry some pretty horrible diseases. Obviously, we know things like rabies, Nipah virus, Hendra virus. We think SARS coronavirus 2, which causes COVID-19, other coronaviruses, and Ebola viruses. But what we don't really understand is why bats are so good at carrying these diseases. And this has been for a few reasons. I mean, one, it's difficult to do work with bats. There aren't that many labs in the world that can actually work with live bats within their facilities. We also have a difficulty with trying to get different types of reagents or testing kits to be able to use on bats that are specific for those animals. So our understanding has actually been, I think, fairly rudimentary in terms of how bats play into this role. But the problem for us is that bats unfortunately are pivotal for us being able to combat many of the worst infectious diseases, or at least many of the worst emerging viruses that we see. And ultimately what can happen is that those viruses can spill over, in some cases, to what we call intermediate hosts. So we think about things like SARS coronavirus. This goes back to this idea of wet markets and in animal markets. Some animals can actually be infected by these different viruses when they spill over from bats, but they don't show the same level of disease that we as humans do. So a good example of this is when we look at MERS coronavirus, so Middle East Respiratory Syndrome coronavirus. At some point in the past, bats basically came into contact with camels, and that virus spilled over into camels. Now that MERS coronavirus in camels, I know that this is a pig, but let's just pretend for a second. MERS coronavirus in camels actually produces a cold-like illness, kind of like what some of the more mild coronaviruses do in humans. So they essentially get the sniffles and get a runny nose. Well, what we think happened is that at some point, that virus basically gained the ability, as it was circulating in camels, to jump over from that intermediate host into humans and cause a pretty devastating disease that kills about 30 to 40% of the people that it infects. So really what we're trying to understand is the dynamic of how all these interactions occur in real time, but at the same time trying to understand how all of this happens at the molecular level. So, how does the virus spill over from an animal? But then when it gets inside of us, what happens when it infects a cell? How does it basically, usurp or undermine and trick our immune system to be able to allow itself to create, millions upon millions of copies of itself and cause, fairly devastating disease in many cases. So my work now pretty much, spans most of Central Africa. I will always have a program in West Africa, and I'll kind of get to some slides for the reason being. But I have programs in Sierra Leone. I'm working in Gabon as well as in Republic of Congo, and just basically started up some new collaborations with folks out of UC Davis in Rwanda to be able to look at gorilla populations there. And of course, we're trying to do work in the DRC as well. And ultimately, again, it's this idea of trying to look at the interface of humans with different animal species, but also taking into account the effects of the environment surrounding that. So, where and how did I kind of get into COVID-19, and why do people maybe seek me out for media interviews? Listen, I'm still trying to understand a lot of that myself. I appreciate it, but chalk it up to growing up in Saskatchewan for trying to remain as humble as possible. But really, my interest in the idea of emerging infectious diseases as it relates back to, say, building capacity at the local level in vulnerable regions, and as well trying to understand how we can better identify methods to treat and provide support for patients. That all came about in 2014. So I was in Frederick, Maryland, so about 40 miles north of Washington, D.C., and we knew that the West African Ebola epidemic was picking up steam. The U.S. government at that point asked for people to volunteer to go and provide direct support on the ground in Liberia, which was at that point one of the hardest hit countries during the West African Ebola epidemic. So I volunteered at the start of August, so the end of the first week of August 2014, and I was on a plane 3 weeks later, which was actually my first trip to Africa. So I went from basically the Integrated Research Facility in Frederick to the Liberian Institute for Biomedical Research, which was actually a malaria diagnostic center, but had been retrofitted to be able to look at Ebola samples. I showed this figure on the right because that's really what we're looking at when we think about emerging infectious diseases in resource-limited regions. So this is one of the first pictures I took when I got to Liberia. This is LWA3, so this is the largest tent hospital site that was set up in West Africa for being able to combat Ebola virus disease. And these are patients that are waiting to get access into LWA3. So this was the first week of September. Beds were already full within that facility. And what happened daily as we drove by this facility daily to go to our own diagnostic facility is that we saw more and more people that were lining up. The unfortunate side was that many of the people that were lining up would die by the time they were getting close to being admitted. But we also saw that many people that had early symptoms of disease or people that were loved ones that were caring for people that were ill were also in fairly close contact with these patients. So it really kind of created a pretty awful situation in terms of disease transmission. But it harkens back, like I said, to this idea of where we're kind of missing the boat in terms of being able to combat diseases in different regions. So I basically had my team that was getting blood samples from around Liberia, and it was a team of a couple of Americans and myself, but as well Liberian researchers who just did an amazing job of work with us. And when we think about this idea of how to combat infectious disease, a lot of times I try to contrast this with our current situation in Winnipeg, in Manitoba, where we're reducing social distancing measures and everybody has kind of gotten tired, of this idea of having to remain isolated or remain locked down, to what basically living conditions are in different areas of the world. So during an Ebola epidemic, these are the things that we're combating, and these are the same things that they're combating right now with COVID-19. Most people are living on a day-to-day wage. So these are actually locals that were coming to our lab to sell us fresh game that they had hunted. That included dried bat, which on these plates that they're carrying were basically chock full of bodily fluids and blood. So we think about trying to combat emerging infectious diseases. We have to understand that people are obviously cash strapped, but there also could be some facilitation of disease transmission through kind of their daily regimens. In Liberia in particular, and this goes for most of West Africa, illegal taxi services predominate in the communities. So a lot of people basically will just take their own personal cars and use these as illegal taxi services. So this is a highway. So these people are driving, I think the speed limit was 60 miles an hour. The car is full of people on the inside. This car has one person sitting in the trunk. We usually would see 2 to 3 sitting in the trunk, with motorcycles working as taxis. We would see as many as 4 or 5 individuals that would be on a single motorcycle. And this is during an epidemic. So these practices, in talking to folks that I collaborate with still in that region, there's no ability to really try and restrict this at the local level. So without having infrastructure in place or funding mechanisms to try and combat this, when we think about things like COVID-19, which are able to spread, between people in close proximity, much better than Ebola. This is what our concern has been. And of course, when we think back to the proximity to wildlife, can't really see it, but in the background, there's a little primate here. In Liberia, what would happen is that we would have a lot of local children that would sell bananas and different foodstuffs on the side of the highway, but they would always have wild animals with them that they had kind of pseudo-adopted. These animals, though, live in the wild and would basically come out and stand by the kids during the daytime to try and get food scraps and attention. So all of this comes back to this idea of, like, how do we combat this to try and reduce disease spread in these regions? Now, I'm going to contrast really quickly to COVID-19, but one of the things I do want to mention when I get into discussing misinformation during COVID-19 and during the pandemic, what I want to get across is that this is not something that is brand new. If we go back to to smallpox in the late 1800s, we go back to Spanish flu in 1918 and 1919. We fast forward now to Ebola in 2014. Misinformation has always been around, and it's always been guided, or I guess it's always used media to its advantage to try and get some of these messages across. But what we do know is that we still haven't figured out a good mechanism to battle misinformation. And all of these things have ultimately resulted in us not being able to potentially curb transmission of infectious diseases as quickly as we possibly could. Okay, so let's get out of Ebola and talk about COVID-19. So coronaviruses, let's just kind of go through a short introduction. I know that I've talked on end about it in interviews, and I know that all of you folks have probably heard about coronaviruses as much as you ever would like to. But I think it's important to kind of highlight a couple of things. So coronaviruses are members of the Coronaviridae virus family. So they're RNA viruses. So their genome is made up of ribonucleic acid instead of deoxyribonucleic acid or DNA. So it's a little bit different than what we see in our own cells in our bodies and in animals. But the genomes of these viruses are fairly large. There are multiple subfamily members, and it gets a little bit confusing when we start looking at the sheer number of different types of coronaviruses that have been identified. But suffice to say, there are multiple coronaviruses that infect humans. So there are in fact actually 7 coronaviruses now that we include SARS coronavirus 2 that can infect humans. And really, from the 1960s up until 2002, people didn't tend to pay a lot of mind to coronaviruses, specifically because of the fact that they really were related back to mild or cold-like illnesses. And we only saw severe respiratory illnesses in people that were severely immunocompromised or people that were quite elderly. And those events happened very, very unoften. But everything changed really in 2002 with the emergence of severe acute respiratory syndrome coronavirus, or SARS coronavirus. And then, of course, changed again with the emergence of MERS coronavirus in 2012. So both of these viruses, have produced epidemics. Are associated with fairly high case fatality rates. So SARS kills around 10% of the people that it infects, so somewhere between 9 and 10%. MERS coronavirus, somewhere between 30 and 40% of the people that it infects. But coronaviruses are not restricted just to humans. We actually see coronaviruses, across any number of different animal species. So that goes everywhere from birds into whales, cows, humans, cats, dogs, pigs. So coronaviruses are fairly ubiquitous in nature, and that makes it a little bit difficult for us because when we start to think about this idea of viruses that potentially could spill over into humans, we're not dealing with 1 or 2 individual coronaviruses. We're actually dealing with a fairly large number. And these are just the ones that have been identified. And I'll show some data later to give an idea of how much broader the coronavirus family actually likely is. So I talked a little bit about this earlier. But when we think about coronaviruses, we prior to COVID-19, we thought mostly about SARS and MERS. And thinking about where things started, the likelihood for both SARS and MERS is that everything started in bats, and at some point in time there was a spillover of either SARS into basically palm civets or other smaller mammalian species that were traded in live animal markets for SARS, or in the case of MERS, that virus spilled over into camels. We think probably somewhere between 3 or 4 decades prior to its emergence in 2012. And what we don't really know is the time period at which these viruses actually circulated in these intermediate hosts before they spilled over into humans. But what we all ultimately know is that whether we're looking at SARS coronavirus or MERS coronavirus, that once that virus got into humans, what we tended to see was that the majority of disease transmission between humans tended to actually happen primarily in hospital settings. So that's kind of been important, right? Is that we did see and we do see some amount of MERS and SARS being spread through direct contact between people in the community, but it largely has been related to transmission within hospitals. And of course, that's quite different now with COVID-19, and unfortunately makes things a lot harder for us to figure out how to combat this. Coronaviruses themselves, for those that are interested in the science behind the molecular science behind coronaviruses, like I am, basically coronavirus is attached to specific receptors on our cells. So if we think about what a virus actually is, I tend to explain viruses as basically looking like a piñata. So we essentially have a hollow shell of protein that essentially is surrounded by basically a layer of fat. And that layer of fat comes from our own cells following infection and following the release of new viral particles back out into or back out to the outside of our cells. And inside this hollow shell of this piñata is basically where that nucleic acid or the genome of the virus sits. So when we think about viruses, they're essentially a big, essentially primarily hollow ball of nucleic acid and protein, sometimes with basically a coating of fat on the outside. So they're amazingly in I guess from my perspective, they're amazingly simplistic. But the problem is what makes them amazingly complicated is that genome. Viruses have been evolving for eons. They've basically been evolving and adapting in the background, and they've basically become perfect microbes for being able to infect very specific types of cells. And in the case of coronaviruses, they have basically antennas on their surface. So on the surface of the actual virus itself, that looks for specific antenna or corresponding antenna called receptors on our host cells. So what that creates is basically a very specific mechanism through which the virus will actually seek out very specific types of cells when we get infected. So the virus finds its cognate receptor on the host cell. That leads to the virus being basically sucked up by the cell. And once it gets sucked up into the cell, the virus actually opens up and releases its genomic material and its genome into the inside of the cell. So at that point, what happens is the virus actually uses our own host cell machinery to essentially create new viral particles. It's kind of like if listen, I'm not a car guy, even though my dad owned a car or an automobile repair shop for years. I could not tell you in most cases, why my car is not working one day. I'm just going to try to keep restarting it. But what I know is if I take my car into an automotive place, basically I'm going to use basically the machinery inside of the automotive dealer to actually repair my car and get it into working condition. Well, viruses do the same thing. They don't need to carry all of the different machinery to create new copies of themselves. They actually can use our host cells against us to do that. And they do that amazingly efficiently. So I'm not going to go through all the different steps, but ultimately what happens is that the virus infects our cells. Our cells basically create new viral proteins, which ultimately results in the formation of new viral particles, and those get released back out of our cells. So this process is occurring basically in the background of infection when we come into contact with coronaviruses. So let's talk about COVID-19. So COVID-19, basically, the first reports came out about COVID-19 starting on December 31st, 2019. And there was basically these initial reports of a SARS-like illness that had emerged in basically what they called a seafood wholesale market in Wuhan, China, so in Hubei province. This wholesale seafood market actually also housed live animals. So that was, one of the first things that we kind of had to work our heads around a little bit. But they did have a fairly broad number of animals that have been related to different coronaviruses in the past at that site. So what was reported on December 31st was that there was these atypical pneumonia cases that had shown up in hospitals in the Wuhan region, and they referred to it as being SARS-like. And I think for infectious disease folks and for emerging virus folks, when you hear SARS-like illness, that gets people pretty concerned because SARS emerged in 2002 and then disappeared in 2004. And we have not seen a reemergence over the course of 16 years. So this, starts to get some alarm bells going. And in fact, listen, I'm very fortunate. We have a 20-month-old daughter at home. But I also have, the other Dr. Kindrachuk, who is very tired and was very tired on New Year's Eve 2019, following her mat leave and going back to work and having our very rambunctious daughter kind of take over our lives. So, kind of fortuitously, I ended up on social media, on Twitter, on December 31st. And saw this report coming out about this SARS-like outbreak, or what they were calling at that point a suspected SARS outbreak in Wuhan. And I was conversing with Dr. Megan May, who's an emerging virus expert in the US, in Maine. And Megan and I had talked about the fact that, this didn't seem like SARS. It didn't have that feeling of SARS, and my kind of initial inference was maybe this was something new for influenza. Maybe it was H7N9, which we knew was present in the region, but it didn't harken back to that. And the reports seemed to suggest that this was something new. And this was, Megan and I were not the only people in the world that were talking about this on December 31st. This was starting to reverberate fairly quickly through the research community. And, ultimately what happened? Well, we know where we are now. So cases have spread across the globe amazingly quickly. So basically throughout January, spreading throughout Asia, and then ultimately as February and March rolled along, spreading across Europe, ultimately to North America and seeding South America as well as Africa. So where are we today? This is where we are. So, I think back to where we were. So I spent 3 weeks in Kenya in January running a science outreach program. And I remember looking at the numbers at that point and thinking, okay, this is not looking great, but we still have a chance to get this under control. And as basically we moved into February, we saw a bit of a lull period. And I think in some cases, people started to think that we maybe had dodged a bit of a bullet. And ultimately what we saw was that cases started to explode in Europe. And obviously, we now see what's going on in throughout the US and South America, but as well now through the Eastern Mediterranean, as well as into the Southeast Pacific. So, we've done very well in Canada, but we are nowhere near out of the woods yet. And this thing continues to grow. And I believe yesterday was actually the highest number of new cases that have been reported worldwide throughout the pandemic. So what do we know? Well, I look at this as being the time period where scientists and researchers and clinicians have really been able to shine. I feel that in the past 6 months, we've gained about a decade's worth of information on a brand new virus that we had never seen before. So within basically the first couple of weeks, that this virus was reported, so December 31st, so by basically the end of the first week of January going into the second week of January, we already had the genome sequenced for the virus, and we were able to basically identify that this virus likely had emerged from bats. And that was really critically important for us because it gave us an idea of where this virus likely was coming from, and ultimately as well started to give some information for how to try and curb other spillovers. And as well, it gave us information for how this virus might behave based on what we saw with other bat coronaviruses like SARS and MERS. So what do we know right now? Well, SARS coronavirus 2, which is what causes COVID-19, has about 96% identity in its genome sequence to another bat virus. So that's why we think that there's a very high likelihood it emerged from bats. But we also see some similarities in the sequence to pangolin coronaviruses. And we've all seen pictures of pangolins. Again, I think they're unbelievably cute, but they harbor and can harbor a number of horrible infectious diseases. But we don't know what role pangolins play yet. So we think that they may be a potential intermediate host. So they may basically be kind of what we think of as an amplifying vessel for the virus that may help facilitate that transmission to humans, but we don't know. What we also have seen in more recent months is that we know now at this point, humans can transmit the virus to multiple different animal species. So we've seen human to cat transmission. We have seen some information suggesting that humans can pass the virus to dogs. And we've also seen human to mink transmission. The unfortunate side is even though with human to cat and human to dog transmission, we haven't seen transmission back to humans. In the case of mink, there's a growing set of data that suggests that mink can actually pass the virus back onto humans. So this actually complicates the story for us a little bit in terms of how to figure out how to combat this virus as it's spread across the globe. When we think about infection, what do we know? So again, going from the standpoint, we didn't know what this virus even was on December 31st. What we now know is that infection occurs in the epithelial cells that line our respiratory tract. So basically the virus looks for a specific antenna on cells called ACE2. It's a specific protein that is found on different cells in our body. During infection, what happens is that the virus gets taken up into these epithelial cells, and the epithelial cells actually have their own immune response. So they actually release different inflammatory molecules, so things like interleukins that signify to the rest of the immune system that the cell is infected. But they also release other things that we call danger molecules that will also basically activate the immune system or the immune responses in surrounding cells. So basically, there's kind of this trench warfare that goes on between viruses and cells at the level of the molecular components within our cells. And this is really how we combat infectious diseases. But we ultimately know that when our cells basically these epithelial cells in our respiratory tract get infected, we can kind of have one of two potential consequences. That really comes back to either mild or even asymptomatic infection versus moderate or severe infection. And we're getting a better picture of what that looks like. So in mild or asymptomatic infection, what we think happens is that our epithelial cells that get infected are actually able to recruit other immune cells very quickly to that section of the lung. Those immune cells basically clean out the virus that's floating around. But that doesn't happen in every individual. In particular, people that have what we call dysfunctional immune responses. So people that are immune compromised, people that are elderly or are battling other infections. What we tend to see is that their immune responses just don't work as efficiently. And we don't specifically know all the reasonings why that happens. But what we think occurs is that in these, this section of the population, basically what we get is essentially an over-amplification of the immune response. So not only do we have, the virus causing basically our cells to respond and over-respond to the infection, but those molecules will ultimately lead to recruitment of a lot of other inflammatory or a lot of other immune cells into those sections of the lung that basically exacerbate that inflammatory response. And that can cause a number of different complications. So I think we're still at somewhat of an infancy in understanding all of the specific mechanisms for how this occurs, but we're getting a much better gauge on what differentiates mild versus moderate or severe infection. We also have a better gauge on how to compare this virus to other viruses. So, as researchers, we sometimes we make I don't want to say we make statements, but we make comparisons that maybe don't come across the way that we intend them to with the public. And I think some of that happened in January, where there was discussion about COVID-19 in China and what we were seeing in terms of symptoms. And there was a lot of discussion and comparison back to influenza. And part of that was to try and quell people's fears about whether or not this was actually going to become a pandemic. At that point, there was still some belief that it could remain localized in China. But there was a lot of discussion about, well, if we look at the seasonal toll of influenza, seasonal influenza kills 500,000 people per year. So if we think on the grand scheme of things, we have to keep in mind that seasonal influenza is also a really horrible disease, yet we don't tend to pay it a lot of attention. And I think in kind of having those discussions, in some cases, people felt that that COVID-19 was actually not that big of a deal. We fast forward a few months, and what we now understand about COVID-19 is that it actually has a number of factors that make it much more difficult to combat than influenza. So first of all, when we look at the number of people that can be infected from an infected individual with either influenza or COVID-19, so specifically seasonal influenza strains, basically one infected person with seasonal influenza can infect approximately 1 to 1.3 other people surrounding them. COVID-19, the numbers are still varying. So we think that they're probably somewhere between 2 and 3. But we also know that there are people that are actually super spreaders for COVID-19. So these are individuals that can actually spread the virus to a much broader range of people. And we think back to this example of the choir practice in the US, where we had one individual that ultimately seeded dozens of different individuals within that choir practice with virus within a single choir practice. So these things are factors that differentiate COVID-19 from influenza. The incubation time for the virus is very different. For influenza, it's 1 to 4 days. So within the time period that you're infected, you're likely going to develop symptoms within 1 to 4 days post-infection. With COVID-19, it's much broader. It's anywhere from 1 to 14 days with an actual mean time of about 5 to 6 days. Well, that makes things a little bit more complicated, because now when we think about people that are exposed or potentially exposed, those people basically could be pre-symptomatic out to the point of up to 14 days post being infected. But we also know now that the virus may actually be being transmitted from people that are pre-symptomatic. So in that period between being infected and developing symptoms, they may be able to expose others to virus. Well, since that's such a broad range of time, and in a lot of cases we've been, trying to dedicate our testing based on the development of symptoms, this makes it really difficult to try and curb transmission at the source. Hospitalization rates are much higher for COVID-19, so about 19% at the time of the writing of this article, or from this data from CDC and WHO, as well as from NIH. So about 19% as compared to 2% for influenza. And the case fatality rates, listen, they're up. I'm not going to make much more of a statement on this, aside from saying that the case fatality rates, I think, are still fairly undetermined for COVID-19, and I think are fairly region specific. But what we do know is that we're seeing a growing number of fatal infections across the globe. Well, we also talk about the idea that there's a massive delay when we think about the idea of doing a diagnosis. So somebody going in and actually getting a diagnostic test for COVID-19. Well, what we know based off of this prior incubation time of 1 to 14 days, most people are likely not going to go in to get a diagnostic check until the point that they show symptoms. And obviously that was what has been mandated up until this time point for people. So the problem is that we also know that people can actually start transmitting the virus prior to getting that positive diagnostic test, and that prior to developing symptoms of disease. So the problem is that there's actually this delay between really when a person can actually start to spread the virus as compared to when they may actually show up as being positive on a diagnostic test. And this actually, again, makes it very difficult for us to kind of assume where we currently are in terms of the spread of infection through the community. This data coming from the US CDC, I think says a million words in one figure. We talked a lot in January about the fact that cases in China largely seem, or at least severe and fatal cases, seem to be in people that were elderly or people that were immunocompromised. I think the problem is that doesn't really do justice to the problem that we deal with COVID-19. And I say that as a simple virologist, so as a non-clinician, so somebody who's not working on the front lines. I think what we have to understand is that for our healthcare workers, they're dealing with a truly unprecedented infectious disease. So we look at hospitalizations across age groups. What we see is that people that are 20 and above basically have fairly similar rates of hospitalizations. So yes, severe disease and fatal disease, it seems to still focus primarily on people that are elderly and people that are immunocompromised, but hospitalizations actually occur across all age groups. So when we think about this idea of the toll on the healthcare system and the toll on our healthcare workers, it's not just because of people that are in these higher age groups. Those hospitalizations occur across all age groups. So when we think about trying to do messaging for people and combating COVID-19, these are the messages that I've really been trying to get across. But what do we not know about COVID-19? And there's still a lot. I will attest to that. One of the key questions is still, when did this virus actually spill over? So this graph is a little bit complicated. It came from a study in Lancet in January, so one of the first clinical cohort studies in China. But what they were doing was basically looking at that initial cohort of people from the seafood market that had shown up in the hospital, and they tried backtracking to see when those people first developed signs and symptoms of illness, and how that correlated back to the seafood market. So I don't want you to focus necessarily on all of these, aside from to see that these are obviously dates going backwards to December 1st. But right here on December 1st, there is a case of COVID-19 that had no contact with that seafood market. So already in January, what we knew was that this virus had likely been circulating prior to December in Hubei province in China. And ultimately, some of those cases, or at least that case, was not linked to the seafood market, which meant that we couldn't just focus all of our resources on trying to figure out what animal may have spread the virus at that seafood market. Now, there could have been people that spread the virus at the seafood market, which is what we think occurred, but we think that the likelihood is the virus was circulating in the community prior to that. And there was new data that was released in a preprint the other day that actually was looking at satellite data and suggested that there was actually an increase or a spike in traffic at different hospitals in the Wuhan region, basically in early to late fall. Of course, all this needs to be validated as to whether or not this correlates with increases in things like influenza-like illness in those hospitals, but it suggests that maybe there was actually something going on in the background. The route of transmission, we think, is still primarily through respiratory droplets, but we have seen instances where there's been data to suggest that things like feces or urine may actually be at least what we call PCR positive for the virus. So those bodily fluids are basically show that the inside of the piñata is present in those fluids. We don't know if the whole piñata is there, which is what we need to infect ourselves, but at least a portion of the pinata is. It's kind of like a fingerprint. So we don't know if the individual that matches the fingerprint is there, but the fingerprint tells us that they might be. And so we're getting some better data on that now. And that's been some of the discussion about this idea of things like fecal to oral routes of transmission, in particular when we think about kids, or in my case, our toddler, because we know that young kids don't tend to show a lot of disease symptoms. So it's difficult to say whether or not they're infected. What do we know about protective immunity? Right now, we're starting to get a better picture of what this looks like. We know that there are indications that people that recover from disease will develop antibody responses. We don't know how long those antibody responses last for and how protective they are, but the data suggests that, at least from what we can see, it looks like what we would expect, that there's some level of immunity that likely is generated, but that still needs to be ultimately validated in much larger subsets of folks. So we can't yet say whether or not, if somebody is antibody positive, so if they do a serology test and they're positive, does that mean that they have protective immunity? I just don't think we're there yet. Does reinfection occur? Everything that we know so far suggests that no. People cannot be reinfected. Most of that comes back to basically data in non-human primates that have shown if you take monkeys that are infected with SARS coronavirus 2 and they recover from COVID-19, if you try to reinfect them, they can't be reinfected. We think that some of the positives that have shown up after people have been negative for longer periods of time likely has to do with sensitivity issues with the diagnostics. So in the last couple of minutes for this, I want to kind of wrap up with this idea of distinguishing misinformation. And this really started in January. So there was a paper that was submitted to a preprint server, which basically is a repository for manuscripts that have been written that have not been peer reviewed by other scientists. So this is basically research groups are submitting this work and basically making it open access to the global research community to vet and provide feedback on. Well, a paper came out that looked at the genome sequences that have been made available and very unfortunately said that they had found some indications that there were segments of the HIV genome within that virus. Now, what I can say is within a couple of days, the backlash on the flaws in the analyses caused the authors to withdraw this paper. So they pulled it from publication. Christian Anderson and his group have done an unbelievable amount of bioinformatics work to prove unequivocally that this is not a bioengineered virus. The unfortunate side is, 5.5 months later, we are still battling with this. I cannot tell you the amount of angry tweets that I get on a daily basis about people that have basically taken this paper and run with it and actually feel that the withdrawal of this paper feeds the conspiracy. Well, what we can say when we think about coronaviruses have been circulating in China for ages. And in fact, Peter Daszak and EcoHealth Alliance just published a paper very recently where they showed that in their work, they identified more than 700 new bat origin coronavirus sequences across China. Now, that's pretty astounding considering that we knew of a few dozen, not more than hundreds. But his work also showed that villagers in Yunnan province that were living in close proximity to bats that carried coronaviruses, those villagers actually showed about 3% seropositivity for bat coronaviruses. So what that means is that in 3% of the villagers that were tested, bat coronaviruses had actually spilled over and caused those villagers to basically generate antibody responses. So coronaviruses are spilling over continually. The problem is that once in a while with SARS, with MERS, with SARS coronavirus 2, you get a virus that spills over that's actually able to cause infection and is able to transmit between humans. One of the last ones that I'll highlight is the 5G conspiracy. So there's actually a pretty strong group of people across the globe that believe that COVID-19 is actually related to 5G technology and 5G cell towers being put up. And they relate this back to the fact that in 2019, there were 5G cell towers that were put up in the Wuhan region in China. And they've gone so far as to basically look back at different pandemics and outbreaks and say, well, if we look at the correlations with all of the different radio technologies and cellular technologies that have been erected, those have all correlated back with outbreaks of disease. And in fact, just simply looking through the dates don't match up themselves. That being said, I've done multiple podcasts with people that wanted to do interviews about 5G, because we've seen people that have been burning down cell towers based on this belief. Lastly, and I highlight it because I also kind of battle with the anti-vax community on a daily basis. There is, again, a segment of the population that believes that diluted bleach is actually what they call miracle mineral solution. So there's a growing belief that if you basically dilute bleach out in a very specific manner, bleach is actually a cure-all for a number of different diseases. And those range from COVID-19 to Ebola to autism. And when I say that these people undeniably believe in this, I am not lying about that. These people believe in this with their full hearts. And this has been growing in the anti-vax community and has spilled out into COVID-19. The simple fact is, we know bleach is toxic. We know that the use of diluted bleach in kids has led to either severe illness or death. But we still see on a daily basis on Twitter, on Facebook, these types of conspiracies that are being spread across the community. And ultimately what this comes back with, I think partially is, I don't want to say it's not a failure in science, but it unfortunately comes back to one of the problems that we've created for ourselves, is that we have basically an impetus to try and publish as quickly as possible. And the problem that I've seen with COVID-19 is that we've seen a massive increase in the number of COVID-19 related articles that are being released as preprints to the general public. And the problem is that, again, the data is not peer reviewed. So it's not fail-safe. Even when it's peer-reviewed, we know it's not fail-safe, obviously. But in this case, it hasn't even passed the smell test of other scientists. Well, the problem is that once this data gets submitted out and once it gets posted on social media, we're already behind the 8 ball with trying to combat misinformation that may come from generalization of those results. So I think we need to find a better way for being able to communicate that scientifically. Ultimately, the reason that we're doing all of this is we are in a true race to find treatments for this disease. We have a brand new disease for which we have no underlying protective immunity in our population globe. We have no vaccines, we have no therapeutics. So we need to do everything we can as quick as possible to try and find these mechanisms to combat COVID-19 while also not trying to feed those different conspiracies and misinformation campaigns. And obviously, the battle with hydroxychloroquine is a perfect example of where we see now politicization of scientific results. So we are truly in an age where we are trying to deal with many different factors that are essentially locking heads at the exact same time during a public health crisis. So with that, I always have collaborators and people to acknowledge. Obviously, my funding sources for the work that I do and for giving me the ability to have this platform. But I do want to highlight my work with Dr. Allison Kelvin and Dr. Daryl Falzerano from Dalhousie University and Vito in Saskatoon, respectively. I think that what we've created is a pretty great triumvirate of researchers that are highly interested and motivated to work on COVID-19. And I've been so lucky and fortunate to be funded along with them to do some of our COVID work, but also to learn and provide knowledge where and when I can. And with that, I'll answer any questions that you might have. Thank you.
Super. Thank you very much. That was very interesting. We have a few questions that have come through. I'll just encourage everybody to go to Slido to post their questions. So the first one is, what is your view on how transparent or not China is being about COVID-19?
So we went right with the hardest question possible. Listen, I am not going to say by any stretch that China does not deserve a massive amount of criticism. Obviously, some of the information we're starting to glean about the fact that this virus was likely circulating longer than what we know. That there will be long-term ramifications from that. Unfortunately, we're not at a point yet where I think we can actually validate all of that data, and we're in this kind of weird position where we need to not stoke the flames, because we still need transparency to some degree with the Chinese government, especially considering the fact we're not through this pandemic yet. So, what happens if we see a resurgence of disease in China? So a potential second wave or additional waves. We need to basically be working with the global community as transparently as possible to try and not only hopefully save ourselves, but to save as many others as we can. So there have been a number of failures, but I think we're seeing that across the globe in different regions. So there will be a very strong message, I think, delivered to the Chinese government if it's found that there has been a delay in reporting. But I think we can't lose the forest for the trees at this point. We need to do everything we can to combat COVID-19.
Okay, so similarly, there are reports from the US that COVID-19 transmission from asymptomatic cases may be less prevalent than first thought. What is your view on it?
Yeah, so this has been the kind of the bane of my existence the last couple of days. So we know that different organizations are obviously will have kind of missteps in how they discuss things, and WHO has done that a few times, though I applaud them for the work they're doing. They have come back to try and kind of step back the statements that they made. But I think a lot of it comes back to this idea of what asymptomatic transmission means. So when we think of asymptomatic infection, we're thinking of somebody that's been infected but never goes on to develop symptoms. So I agree that asymptomatic transmission is likely not driving the pandemic. It likely occurs, but it probably occurs in a small degree of patients. But that doesn't necessarily correlate to pre-symptomatic transmission. So during that pre-symptomatic stage, that incubation stage of infection, when somebody is not showing signs of disease but they're infected, they don't have any symptoms. So they're asymptomatic, but they will ultimately go out to spread disease. So my concern with the statements is that if we put these things out, what it tends to create is basically a belief in people that if they don't show any signs or symptoms of disease, that they're fine, that they can go out in public. Well, in fact, we know that pre-symptomatic transmission is actually helping drive the pandemic. So, we need to try and still get the message across that for those people that have been in contact with people that have been sick with COVID-19 or are potentially have been in contact, we need people to, again, keep that minimum 6-foot distance between themselves and others, wear a mask if they can't maintain that, and undergo proper hand hygiene. We have to try and figure out how to not kind of shoot ourselves in the foot with our messaging in this regard.
Okay, so how confident are you in reported number of cases? Is this hampered by poorer areas of the world where medical care may not be sought, and so are cases being missed?
Oh, yeah. So really great question, right? And talking to folks in Sierra Leone and Kenya, as well as folks in Gabon, this is a real issue, right? So, again, we have to understand that in these regions of the world, they are facing emerging infectious diseases all the time. Coming out of West Africa, coming out of Ebola, there is still a fear in the public that going to a healthcare facility basically puts you into essentially a 50-50 chance of stepping back out. So there's still that fear factor that if you go to a hospital, you are going to die. And that's one of the issues that we need to, kind of again, probably through the longer term, figure out how to communicate better at the local and regional level with our community partners. But cases likely are being underreported. And we know that even here in Canada, we know that the limitation of testing reagents and testing kits really led us to only be able to test those people that had symptoms. Well, that's probably a very small proportion of the number of people that actually were exposed to the virus ultimately. So, what we're hoping over time is that through things like serology testing, where we go out into the community and look for the development of antibodies, what we'll get is hopefully a better sense of how many people were truly exposed during this first wave of infection. And that will give us a better indication of how many true cases of actual infection there were within the communities. And this will be something that will go on across the globe as well.
Okay, the next question is, what have we learned from different approaches that countries are taking to battle COVID-19? And the specific countries that are mentioned are Sweden, US, and Canada, but essentially all other countries.
Yeah, so I'll use the US as an example first. Not to try and pick on our southern neighbors, but I feel like I have some levity because I lived there for 7.5 years. So I can make some statements. Listen, what we're seeing in places like Tennessee, California, Texas, Florida, what we're seeing is massive upticks in cases very quickly. And what a lot of this seems to harken back to is the reduction in social distancing measures before cases had actually truly plateaued. So I think what we can learn very quickly from that is, first of all, Canada has done an amazing job. I think here in Manitoba, we've undergone reduced social distancing measures at a period in time when it was safe to do so, and hopefully, fingers crossed, the numbers kind of stay where they are. We're seeing other regions in Canada that are following suit, but we haven't had that same push to basically just open everything up en masse. And in the US, I talk a lot about this idea that there's basically been this kind of blending of a public health crisis and politics. We know we're in an election year in the US. So the unfortunate side is that we know the economy is going to likely have an effect in how voter turn or how votes come in for the two different candidates. That unfortunately also probably plays into some of those decisions. And I think we need to figure out a mechanism to try and kind of combat that. With the Swedish example, I mean, folks within Sweden themselves and within the Ministry of Health have said now that they probably underestimated COVID-19. They haven't necessarily walked back to say that they made the wrong decisions, but I think what we've ultimately seen as compared to other Northern European regions is that without basically full physical distancing measures being put in place, what happens is that ultimately you get more people that are dying, and you ultimately get a greater burden of disease. So I think we can look back at some of the failures that we've made nationally as well as regionally with COVID-19, but we can also kind of give ourselves some applause for the fact that I think we've done very well, as compared to the global community.
Okay, the next question is, in your opinion, what is the likelihood that an effective vaccine will be developed? And if so, when?
Yeah. So, I had the great pleasure of walking past Dr. Tony Fauci nearly on a daily basis when I was at NIH. So I have an unbelievable amount of respect for his opinions and his viewpoints. I think that his views on the vaccine or on having a vaccine candidate licensed and out to being deployed of being kind of that 12 to 18 month range is feasible. I think the thing we have going for us is that there are a number of different vaccine platforms that are being pursued across the globe, and we know that multiple vaccines obviously are in clinical trials at this point. So I think that what we're hoping is that maybe some of those timeframes will backtrack a little bit and may actually come into place a little bit sooner. But when we start to think about the idea of not only passing, say, safety and efficacy standards, but also then going through manufacturing, getting basically doses created in millions, and then getting those dispensed out to communities, and then getting people to be able to come in to get immunized, it's going to take a lion's share of effort. So I think we are still going to be in the clutches of this thing probably for again, I would think that 9 to 12 month range at a minimum.
Mm-hmm. Okay, thank you. This is very interesting. This question: will COVID-19 slowly disappear by itself? Can we become immune to COVID?
Yeah, so this is a great question, right? And we look back at the original SARS coronavirus and MERS coronavirus. What we saw with both those viruses is that people that were exposed ultimately developed some level of protection, but that only lasted somewhere in the network of months to a couple of years. So the concern for us with COVID-19, especially with the fact that we are 6 months into this, and we're likely going to be within the clutches of COVID-19 for the foreseeable future, at least through the outset of this year. What we have to start to think about is, okay, what happens if immunity only lasts for a year naturally? So somebody that was exposed in January or February, maybe in early 2021, they're no longer immune protected. So I think for us, we're kind of in that stage where we don't quite know. We saw with SARS coronavirus, the original SARS-CoV, that just through proper containment and contact tracing, once we limited the ability of that virus to transmit, the virus ultimately disappeared, and we haven't seen it spill back over. COVID-19 unfortunately transmits a little bit more widely and a little bit more easily. So that's going to make it more difficult. And I think if anything, the concern right now is that if without vaccines or therapeutics, ultimately, could COVID-19 become something that becomes endemic, like other coronaviruses that cause more mild-like illnesses? So I think the jury is still out. And I think we're too early at a stage to really get a gauge on what this virus is going to do. It's been unpredictable to this point, and it's probably going to remain unpredictable.
Okay, thank you. The next question is, there have been reports that people who've recovered from COVID go on to deal with the linger effects, like lung issues and strange blood work. What are your thoughts on that?
Yeah, my thoughts on this are it's unfortunate, but I don't think it's completely unsurprising, because we've seen this with original SARS coronavirus as well as with MERS coronavirus, and we see this even in my work with Ebola virus disease survivors. We see long-term complications in those folks that had severe disease. So for people that had COVID-19, in particular people that had moderate or severe disease where they were hospitalized, I think we're at this kind of early stage where people that were infected in China in January are now 6 months out from being infected. Well, I guess 5 months out from being infected. We're just starting to get a bit of a glimpse of what the long-term picture looks like in those patients. So the likelihood is, from some of the data that we've seen being accrued from different regions, obviously through Europe and data being released out from China, that there are concerns about potential long-term lingering effects, but we don't quite know what that looks like yet. And at the same time, we're dealing with a virus that has now infected millions upon millions of people. So what we're now seeing are even things that occur in a very, very small proportion of the population. We're seeing obviously larger numbers and a higher representation of that because of the fact that we're seeing so many infections as compared to, say, MERS or SARS. So we may have also seen these types of ailments in those diseases, but they just got washed out because they never spread as far. So I think the likelihood is that, yes, we're going to see long-term complications. We don't know what the picture looks like, but we're getting a better picture as 2020 continues onwards.
And so similarly, it's sort of a follow-up question, is from the point at which a person is infected, how much time must pass before a test for COVID-19 will reliably detect this infection?
Yeah, that's a great question, right? So one of the things that has come out recently is we think, or at least I'll say we on behalf of the research community, we think that basically the highest viral loads, the highest amount of infectious virus that a person is carrying occurs kind of at their peak of illness. So basically at the point of illness where they have the worst symptoms. So what that means for us from, say, things like a testing perspective is that the likelihood is the most reliable test is going to be that which kind of fits that perfect period when somebody has the greatest amount of virus within that sample, which also correlates with the highest amount of symptoms. Now, the problem is that somebody that is mild or mildly symptomatic versus somebody that is severely ill, those things are going to look very different. So the problem that we get into is this, this idea of, well, when do you go to get tested? Well, we know right now if somebody is not symptomatic, even if the virus is kind of incubating in the background, the likelihood is that those people will probably test negative. But that's a false negative. The virus is there. It's just at too low of a level to be picked up by the diagnostic assay. So I think the same recommendations that have been made for the past few months still stay in place. That people that have had close contact with somebody that has had COVID or may have developed symptoms of COVID, those people should self-isolate. And if and when, and obviously that still goes for 14 days at that point. But if they develop symptoms, that's the point where we want to see people asking for tests. And tests will increase in numbers over time as more testing kits become available, but they're still going to focus primarily on those people that are symptomatic first.
Mm-hmm. Okay, thank you. We've got a number of questions still. What do you think might be an effect of intensive livestock operations on possible future spillovers to humans?
Yeah, so this is such a great question, right? And it's something that that we talk about a lot in my lab, and as well with with folks that I collaborate with. So when we think about this idea of people that are in close proximity to wildlife, what we do know is that a lot of different livestock species are potential mixing vessels. So mixing vessels. So we think about things like poultry. We know for influenza viruses that poultry are a mixing vessel for different influenza viruses. Same thing with swine. So we know that people that work in close proximity to that type of livestock, are probably at a greater likelihood of getting infected. Now, there's environmental conditions to take into account, which is why when we look at influenza, the majority of kind of highly pathogenic influenza viruses have tended to emerge in Southeast Asia. With coronaviruses, again, we have we're getting, I think, a better picture. And I think Peter Daszak and EcoHealth's Alliance work in China looking at bats is a perfect example where the more that we start to do surveillance in wildlife, the more that we start to pick up these viruses and we start to figure out, okay, is it all bats or is it actually just specific species of bats in this region because of the environmental factors that come into play? And if we can understand that aspect, what we can start to do is hopefully guide people with their livestock practices. So we think about things like Nipah virus that emerged in 1999 in Malaysia. That spilled over from bats into pigs and ultimately spilled over from pigs into humans. If we can try and increase essentially that distance between bats and those potential incidental hosts, that could essentially amplify the virus and transmit it to humans. Hopefully that will be enough to curb transmission, because ultimately, with these viruses that are found in species like bats, we're not going to be able to eradicate them like we did with smallpox, because by the time we eradicated smallpox, it was only found in humans. So as long as we try to immunize all humans, the virus couldn't basically transmit any longer. With these other zoonotic viruses, unless we can actually cut the virus off at the actual animal itself, the virus is still going to circulate in the background. The best we can do is try and stop that spillover event.
Okay, thank you. The next question is, if RNA is so fragile in the environment, how can it be detected in things like sewage?
Yeah, so this is a great question, right? And it comes back to, listen, thankfully, a lot of RNA biologists are far smarter than me, which is why I rely on a lot of their data. What we know with nucleic acid, though, is that so RNA is fairly fragile. It breaks apart, but it doesn't break apart necessarily down to the single nucleotides. What it will do is likely breaks down into smaller pieces, and those pieces may actually be able to form different types of secondary structures that maybe make them a little bit more resistant to things in the environment and to degradation in the environment. So when we do things like these different types of diagnostic assays, these qPCR assays, what we're really looking for is a small fragment of that genome as opposed to the whole genome itself. So even if we get a positive test, it doesn't necessarily tell us for certain that there's infectious virus there. So like, again, my work with Ebola patients, we look at Ebola virus persistence in the testes of males that survived. And we know that we're able to detect RNA of Ebola virus in those survivors out to, in some cases, more than 40 months in their semen. But that doesn't necessarily tell us that it's infectious virus. So, we're able to utilize basically some of, the resistance to full degradation by RNA to use it as, basically a hint or an alarm bell that there may be something there. But then we have to go basically the extra mile to identify if there is actually that whole live virus or that whole infectious virus or that whole pinata actually in those samples.
Okay, thanks. The next question, going back to politics, is how important in the US was Trump's disbanding of a committee or panel that was planning on how to deal with future pandemics last year?
Yeah, this is how do I say this without without trying to grimace or say anything untoward. Listen, I think it had a massive effect, right? And I, I was lucky enough to be in the U.S. during the Obama years. There were obviously problems with some of their response efforts. We saw that in some of the 2009 H1N1 response. But by the time that Ebola came around, they had a really great committee for dealing with pandemic preparedness. And ultimately, we know that those recommendations were in place with how to deal with pandemics as they would potentially arise, as opposed to trying to deal with them, 3 or 4 months following spread. And a lot of that, again, harken back to this idea of putting focus on vulnerable regions of the world and the importance of putting investment in those regions. So ultimately, listen, my feeling is that it's had a massive effect. And if anything, what we've seen, the effect has really been that there's been a division along political lines with how serious, in many cases, how serious they feel COVID-19 is. And that's the unfortunate side that we don't want to be in when we're battling a pandemic, especially during the early stages, and with something that's unpredictable. When we get people that are divided, and we're seeing further division, how do we then come back if there's a second wave to get people to adhere to public health recommendations, to go back into lockdown or to go back to social distancing? So I think it's had just an amazingly deleterious effect.
Mm-hmm. Okay, thank you. We just have time for maybe 2 more questions. The next one is, how will scientists determine the efficacy, including the long-term efficacy of antibodies in preventing infection in the person who has these antibodies?
Yeah, this is a great question, right? So one of the things that we can do is, there's a couple things. One is we can actually do different molecular tests with antibodies. So if we get a serum sample from a patient, what we can do is we can actually look to see whether or not those antibodies will do what we call neutralize the virus in cell culture. So in my laboratory, you could do basically a very quick test to see whether or not the antibodies that are within that person's serum are actually able to essentially inhibit the virus from being able to infect cells. So those are kind of quick fixes for us. The other thing that we can do is we can actually kind of go back to bringing in animal models of infection. And I know that there is still, definitely in the U.S., a community that feels that we should not be doing animal testing, and I understand to a certain extent their feelings. But the fact is, when we're dealing with an emerging disease, we know that we don't necessarily have, again, that levity to be able to wait to see whether or not people that were infected in this first wave ultimately get infected in the second wave. Well, what if we can show in non-human primates that if we infect them with SARS coronavirus 2, they basically develop a disease that looks very similar to COVID-19. And in fact, if those animals, and when those animals recover, if we go back to reinfect them, weeks, months, or years after they were initially infected, are they still protected? And we can use that again as somewhat of a guide for us with being able to demonstrate whether or not we actually see protection. And again, we can always go back to the communities if we see subsequent waves of infection. If we have data recorded for which patients were initially infected during the first wave through contact tracing, we can look to see whether or not those people or their potential household contacts were ultimately infected again during the second wave. And that again gives us some idea of whether or not they've been protected.
Mm-hmm. Okay, thank you. What is the latest, most reliable information on SARS coronavirus 2 viability on and the transmission via various services versus face-to-face transmission?
Yeah, great question. So I actually had a CTV Winnipeg interview this morning at 7:30 that asked this exact question. So where are we? We're still in the same place we were, probably about a month and a half ago. So the Rocky Mountain Labs in Hamilton, Montana, which is part of the NIH, through Vincent Munster's group, had actually done some pretty amazing work looking at surface stability of the virus. And what we know is that basically the smoother the surface, so things like glass, or ceramic, the longer that virus could remain infectious on that surface. But what we need to keep in mind is that when we start to think about things like environmental factors, so temperature differences, sunlight, rain, wind, all these factors will have basically negative effects on the viability of that virus. So we still think that ultimately probably the 24-hour range is what we're looking at. But what we don't truly know is how much virus you would need on a spot, on a surface, to actually infect somebody. So if I touch my counter and then touch my eyes, how much virus needs to be there for me to get infected? Now that's different from somebody that is within, say, a couple of feet of me and happens to cough, because those people likely, or if they speak loudly, if they expel any kind of respiratory droplet, they're likely carrying a much higher burden of virus on those droplets that I'm going to be exposed to. So what we can ultimately say is that contact transmission likely leads to some amount of infection, but the likely driver of the pandemic is still person-to-person contact.
Okay, thank you, Dr. Kindrachuk. Unfortunately, that's probably all the time we have for questions. So I'm sorry to everybody whose questions we couldn't get to. But thank you very much for your very interesting and very timely presentation. Thank you to all of you who've been listening and participating in asking questions during this last hour and a half. Just a reminder that we will be posting a recording of today's session on our website, as well as sending you a follow-up link to a survey where you can share your thoughts and feedback. And please do provide feedback, as it's the only way that we're able to improve. Next week on June 17th, our speaker will be Dr. Suzanne Gagnon. She is the Canada Research Chair in Leadership Education, Assistant Professor in Leadership and Organization, and Director with the James W. Burns Leadership Institute here at the University of Manitoba, who will speak on crisis leadership observations for a new model. Just a reminder, we do have 3 sessions left, including next week on the 17th. We also have a session on June 24th and a bonus session on Tuesday, June the 30th, all of which are starting at 1 p.m. Central Standard Time. Please do visit our website to register for these sessions if you haven't already through Eventbrite. In the spirit of sharing more Learning for Life opportunities, we will also be sending you an email about this tomorrow, but we're excited to share that on Friday, June the 12th, that's this Friday, you can join the University of Manitoba Faculty of Science at 3:30 p.m. Central Time, where you will hear from Nobel Laureate and alumnus Dr. James Peebles, who will be presenting on lessons on the nature of physical science from the study of the expanding universe. And as I said, we'll be emailing you with some details tomorrow about that. So with that, have a wonderful week and please stay safe. Thank you.